The last speaker I'll be introducing today is Jacquelyn Meisel, and she's a graduate student in Elizabeth Grice's lab at Penn. And her topic is the skin microbiome. Characterizations are that they're biased based on sequencing approach. Welcome. Okay, so all your slides will be there. Next is here, pointer is here, and back.
Oh, great, thank you. So I'd like to thank the organizers for inviting me to share my research here today. Covering almost 2 square meters of surface area, our skin is colonized with a variety of microorganisms, including bacteria, fungi, viruses, and mites. And while most of these bacteria are commensal and not pathogenic, we know that alterations in our skin microbial communities are associated with a variety of different dermatological disorders. In particular, acne is thought to be associated with particularly virulent strains of the skin commensal Propionibacterium acnes. A recent study sampled sebaceous glands of healthy and acne-affected patients on their nose and identified unique Propionibacterium sequences or ribotypes within these samples. And so in this figure, they're showing each column is a different sample, and the colors are representing the different unique Propionibacterium sequences or ribotypes. And we can see that certain ribotypes are associated with healthy skin, shown here on the right, and others, such as ribotype 4, highlighted in pink, are enriched in acne samples. And this ribotype 4 was also found to contain predicted potential virulence factors that may contribute to its pathogenicity. And so ultimately, we envision that we can manipulate our skin microbiota to either treat or prevent skin diseases, but we need to have a better understanding of our skin microbiome before we can get to that point. Culture-independent high-throughput sequencing of the 16S ribosomal RNA gene has been used to characterize the microbial communities living on our skin. The 16S gene is found in prokaryotic organisms, highly conserved, and contains conserved regions and hypervariable regions, here in the gray, V1 to V9. And these hypervariable regions contain species-specific signatures that allow us to identify, to classify taxonomically what are in the samples. And while the acne study that I just talked about sequenced the full 16S gene, this is typically not practical or cost-effective. When you use cost-effective next-generation sequencing technologies, you have shorter sequencing read lengths, so you're often required to select hypervariable regions that you're focusing on to sequence. In a typical 16S study, you start with your microbial community sample, you amplify and sequence the 16S ribosomal gene, and then once you have your sequences, you group these similar sequences into OTUs, or operational taxonomic units, which you can compare to reference databases and identify what your community composition is. Another technique is whole metagenomic shotgun sequencing. And in this case, you're not amplifying a specific marker gene, you're extracting all of the DNA and sequencing all of the DNA from your sample, comparing your sequences to reference genomes, and comparing to different databases to determine the relative abundance of gene pathways in your sample. So not only can you determine what organisms are in your community, you can identify what they're actually doing. And so our lab is interested in optimizing sequencing techniques for skin microbiome studies. We want to address 2 main questions. Because next-generation sequencing technologies require you to focus on a specific hypervariable region for sequencing, and different hypervariable regions of the 16S gene are better at classifying different types of bacteria. We want to determine which hypervariable region is going to provide the best resolution for skin-specific studies. And secondly, we want to identify the additional function that whole metagenomic shotgun sequencing provides when looking at skin samples. Whole metagenomic shotgun sequencing does provide the additional ability to look into functional categories. However, it's a lot more expensive to collect and analyze. And there are computational techniques that now exist that allow us to infer functional profiles from our 16S data sets. And we want to see if these computational techniques are sufficient to capture most of what's going on at the skin, or if there's additional insight provided by shotgun sequencing. In order to address these questions, we collected cutaneous swabs from a cohort of 20 healthy volunteers. We collected from 12 different body sites of different skin microenvironments, so sebaceous sites like the retroauricular crease, moist sites like the toe web, and intermittently moist sites like the palm. We extracted the DNA from these swabs, and in addition to whole metagenomic shotgun sequencing, we also did 16S sequencing of 2 commonly used hypervariable regions in the 16S gene, the V4 hypervariable region and the V1, V3 hypervariable regions, which were used in the Human Microbiome Project. Once we obtained our sequencing data, we filtered out low-quality sequences. We taxonomically identified what bacteria were in our community samples. We calculated the diversity within each sample, and then we used predicted gene content to identify predicted functional enrichment. And we did these analyses using CHIME software, and further analysis was done in our statistical computing environment. As a control, we sequenced a mock community which contained 20 known bacterial strains. So this is the relative abundances of the 20 different bacteria that are contained in this sample. And because we know what to expect to find in our sample, we can use it to determine how accurate our different sequencing techniques are. And so in this heat map, each column is representing a different sequencing type, where actual is referring to what we expect to see in our mock community based on its composition. Each row is a different bacterial genera, and the colors are indicating the relative abundance of the different bacteria in the samples, with the darker purple being higher abundances. And so we can see that our shotgun sequencing data is much more similar to what we expect to find in our mock community. The V1, V3 does a fairly decent job, but it is underestimating Streptococcus and Staphylococcus and Propionibacterium levels, whereas the V4 samples overestimate Streptococcus and Staphylococcus and severely underestimate levels of Propionibacterium. This is looking at our actual data we collected from our skin swabs. Each box is a different body site, and the 3 columns are different sequencing methods, V1, V3, V4, and whole metagenome. And if we use our whole metagenome samples as kind of our control, we can see that our 2 16S methods are underestimating levels of Propionibacterium, shown in the dark blue. And unsurprisingly, moister sites like the toe web and umbilicus that have less Propionibacterium are more similar across the 3 sequencing methods, whereas oilier sites have higher differences between the 3 sequencing methods. We next looked at 3 of the most common skin commensals, Propionibacterium, Staphylococcus, and Corynebacterium, and we correlated the relative abundances from our 16S samples on the y-axis and our whole metagenome samples on the x-axis. And we can see the V1, V3 samples on the bottom row have stronger positive correlations with the whole metagenome data set than the V4 samples do. Because of the short sequencing reads that we use for our 16S sampling, we're often unable to classify things at the species level. And so we were able to use a robust Staphylococcus database and a phylogenetic placement program called pPlacer to identify Staphylococcus species in our 16S samples. So these pie charts here are showing all of the sequences that were classified at the genus level as Staphylococcus in our V1, V3 data set and our V4 data set. And the dark purple is indicating those that we were able to classify further at the species level using pPlacer. And so you can see almost 62% of the V1, V3 staph sequences were classifiable at the species level, but less than 1% of the V4 samples were classifiable at the species level. And then these bar plots are looking at the sequences that were able to be classified at the species level. What were they? So each bar is a different body site, and the V4 samples contain a lot of Staph aureus, whereas the V1, V3 samples have a lot more Staph epidermidis in blue and Staph hominis in green, which is more comparable with what we see in our whole metagenome samples. Next, we looked at alpha diversity, or the diversity within each sample. And we use the Shannon Diversity Index, which takes into account both richness, or the total number of species in your sample, and the evenness of your species, the species that are present. And so we can see here our 16S methods, V4 in blue and V1, V3 in red, are estimating higher diversity within our samples compared to the shotgun sequencing data set in green. Not only that, while all three methods find sebaceous sites to have the lowest amount of diversity, the 16S samples are predicting intermittently moist sites to have highest alpha diversity, whereas the whole metagenome samples find moist sites to be the most diverse. And then finally, I predicted or calculated functional enrichment in my shotgun sequencing data set. I inputted my quality filtered sequences, blasted them against the KEGG functional database. I annotated them with a program called Human, which generates a relative abundance table of different KEGG pathways within each sample. And as I mentioned earlier, computational techniques now exist that allow you to predict gene or functional capabilities from your 16S taxonomic data sets. So we used a program called PyCrust, which uses reference databases to pre-compute gene content for each OTU, or operational taxonomic unit, or basically each species. And you can input your OTU table with your counts per each sample. The program normalizes it by predicted gene copy number, infers metagenomic content, and outputs a relative abundance table of predicted KEGG pathways. And so in these plots, we looked at the V1, V3, and V4 samples are in blue and red, and the whole metagenome profile is in green. And we can see that while for most KEGG pathways, there is a fair amount of agreement in abundance, certain pathways such as membrane transport are enriched in the 16S predicted functional profiles, whereas others such as metabolism of cofactors and vitamins is enhanced in the actual whole metagenome functional profile. And so in conclusion, we found that whole metagenomic shotgun sequencing provided the best resolution of our mock community samples. We found that sequencing the V4 region of the 16S gene fails to detect common skin commensals, especially Propionibacterium. We found that taxonomic alpha diversity trends varied by sequencing method we used, with our 16S methods underrepresenting the diversity at moist sites as compared to our shotgun sequencing data sets. We found that our KEGG functional profiles varied by our sequencing method. And while we found shotgun sequencing to yield the most accurate profile of our skin microbiome, we did find that sequencing the V1, V3 region to be a good cost-effective alternative. So I would like to thank everyone who contributed to this project in Elizabeth Grice's lab, as well as my funding sources. And thank you for listening.
Are there any questions?
Hi, that's really nice. I'm curious regarding the issue of counts and using PyCrust to determine the presence of functional pathways. You use the number of reads and counts to be able to determine how much of a particular pathway is present. How do you grapple with that, given at least my understanding is counts from sample to sample, since they're all kind of relative based on one sample? How do you determine that when you're looking across samples?
So we subsampled all of our sequences to 2,500. So they were the counts should all add up to 2,500 per sample. So I don't know if that answered your question. Okay, I can talk to you later, yeah.
Hi, that was a great talk. Two questions. First, could you comment on how the samples were collected physically? And then secondly, can you comment on the sensitivity of the whole genome sequencing technique?
Sure. So the samples were collected with like Q-tip swabs and yeast cell lysis buffer, and you just swabbed a particular area. What was your second question?
The sensitivity of the method. So assuming that all three methods you were comparing the bacteria were collected equivalently, is there a greater sensitivity of one method versus the other in terms of how few genomes need to be there in order to be detected? Do you know how many bacteria were in your mock samples?
I'm not sure. For the whole genome sequencing, we used a Nextera kit where it was less than a nanogram of DNA is required. I don't know actual bacterial counts. Like we just we extracted the DNA and sequenced the DNA. So
the DNA, the minimum level of DNA needs to be nanograms or picograms, or?
Yeah, for the whole metagenome sequencing, it's the Nextera XT kit that we used requires less than a nanogram.
Okay, thanks.
Thanks.
Thank you.
Thanks. I had one question about the terminology at the beginning that you used. With the methods that were done with the ribotyping, where the study you quoted that looked at the ribotypes 4 and 5, those samples were taken of all of the contents of the follicle, with a strip. So it's really not sampling the sebaceous gland, or it's not sampling a sebocyte. It's sampling all the contents in the follicle, which are mostly keratinocytes, as well as sebum and bacteria and whatever else is in the follicle. But the actual sebocytes themselves are not removed by doing those types of sampling techniques. But that was a great talk. You did a great job.
Thank you.
Thanks.
From the macroeconomics and practice gaps of treating acne and rosacea to the microbiome, from the giants of dermatology and acne and rosacea, like Dr. Eichenfield and Dr. Thibotot, to undergraduate students, I think that we've covered a broad range of topics with acne and rosacea and learned a lot today. I thank the American Acne and Rosacea Society for organizing this event again, particularly our executive director, Stacy Moore, and my co-chair, Dr. Diane Thibotot. I've really enjoyed learning today with our shared interest in this topic. Thank you so much.